Okay, good morning. Thank you very much for joining our DiscoverX webinar. I think we would like to get started with today's presentation. Unfortunately, we cannot answer any questions in real time. So if you have any questions regarding any of the materials presented or anything that you've read in the past about BioMAP and DiscoverX technologies, please go ahead and find the chat or the Q&A windows that come with the software. And if you type in your questions there, we will make sure to address those questions at the end of our presentation today. So today I want to introduce a very, very good friend as well as a great colleague, Dr. Charlene Velichko, who's our Director of Research Biology. And today she will be talking to us about our BioMAP technology platform with the presentation today, combining target-based and phenotypic discovery assays for drug repurposing. And this has been a very, very hot topic, not just in North America and Europe, but many of our inquiries from Asia Pacific have really talked about how do we reuse our current assets or reposition our current assets. And BioMAP is actually very, very well positioned for this. So without further ado, I'd like to introduce Charlene.
Thank you very much. Okay. There has been much discussion around target-based versus phenotypic approaches to drug discovery. My message today is that it doesn't have to be one or the other, but that in fact the two methods are quite complementary. Today, I'll be showing you an example of how information taken from both approaches can be used to prioritize known drugs for repurposing for a specific indication. The cost of bringing a new drug to market today is astronomical, estimated to be, on average, well north of $2 billion. Additionally, the failure rate is high. Only 15% of compounds that enter phase 2 clinical trials are ultimately approved. So with that in mind, drug repurposing programs are an attractive way to either expand the indication space for an existing approved drug, also known as repositioning, or salvage compounds that have failed clinical trials for their primary indications. In addition to expanding the number of patients that can potentially benefit from these drugs, the time to approval is generally much shorter, a 3 to 5 year range compared to a 10 to 20 year range for new chemical entities, partially due to an established safety protocol. Launching a new drug repurposing or repositioning program starts with indication selection. Although several successful drug repurposing programs have been based on serendipitous discovery or by pursuing disease indications with highly similar pathways to approved indications, for example, psoriatic arthritis and rheumatoid arthritis, evidence-based guidance on indication selection can streamline this decision-making process and expand the pool of potential therapeutic areas, thus increasing the chances of clinical success. So many diseases have a complex pathophysiology. An example of this is idiopathic pulmonary fibrosis, also known as IPF. IPF is characterized by progressive scarring of lung tissue, which eventually leads to the inability to exchange oxygen. Years of academic research have identified many molecular targets or pathways known to play a role in IPF. For example, TGF-beta signaling, TGF-beta signaling, ER stress, and macrophage biology have all been well documented as potential disease drivers, but it's unlikely that we have all the pieces to the puzzle. And like a three-dimensional puzzle, each piece influences how the other pieces fit in and determines the overall final shape of the puzzle. Target-based approaches focus on these known pieces without a full understanding of a drug's impact on a larger puzzle. In addition, single target-based approaches do not take into account the potential benefit of polypharmacology, which may make significant impacts for a multidimensional disease such as IPF. In fact, the two approved treatments for IPF, pirfenidone and nintedanib, are thought to be polypharmacologic in nature. Phenotypic approaches allow what is known about the disease to define the three-dimensional shape of the puzzle. By combining phenotypic and target-based approaches, one can help refine and characterize the pieces of the puzzle. The BioMAP platform consists of human primary cells that are co-cultured in vitro and stimulated to model complex disease biology. These cultures, which we call BioMAP systems, are initially validated using multiple drug or tool compounds, and then the culture conditions and the readouts are locked down and tested repeatedly to show robustness and reproducibility. Systems are grouped together in panels. The effect of compounds on clinically and biologically relevant protein-based readouts are measured across multiple concentrations and displayed as a profile. This profile essentially serves as a phenotypic fingerprint of a compound. The BioMAP platform incorporates a suite of tools that can be used to address the needs of drug repurposing programs. And the main tools are panels, which cover broad disease biology, allowing for the simultaneous evaluation of compound effects on diverse tissue and disease biology, as well as disease-focused panels for fibrosis, oncology, and autoimmune disease. Because we run these systems in a robust and reproducible manner, it has allowed us to build a large reference database of BioMAP profiles. We have been able to develop a rich knowledge base and a suite of informatic tools to interrogate this data. An example of this are the signatures of safety and toxicity that we've been able to develop based on BioMAP profiles of compounds that have failed clinical development due to adverse effects. I won't be able to touch on this today, but this platform is also useful for the systematic evaluation of drug combinations. Combinatorial approaches are being heavily investigated for drug repurposing programs. As I mentioned, we've built a database of profiles from about 4,500 reference compounds. This database contains approved and failed drugs, as well as tool compounds that are small molecules, as well as biologics representing many different target mechanisms. This information is valuable in identifying potential mechanisms of action, comparing activities to gold standard drugs for a particular indication, and establishing biomarker profiles associated with efficacy and safety. Our goal in this exercise was to use a fairly straightforward approach to identify known drugs that may have the potential as an IPF treatment. The first step was to determine the phenotypic signature of the two existing approved therapeutics, pirfenidone and nintedanib, and to find their shared effects as a signature of a clinically successful IPF drug. To go back to our original analogy, we are using these two drugs to outline the shape of the puzzle. As I mentioned, these two drugs are thought to be polypharmacologic in nature. The target or targets of pirfenidone are not completely known. Nintedanib is a multi-tyrosine kinase inhibitor, best described as inhibiting the signaling of angiokinases such as VEGF and PDGF. Outside the U.S., nintedanib has also been approved for the treatment of non-small cell lung cancer. Pirfenidone and nintedanib are not thought to share any targets. But assuming that they do share phenotypic activity, we then use the shared signature as a scorecard to systematically score the compound profiles of known drugs in our reference database in order to potentially identify novel drugs or target classes that share this signature. We then evaluated the activity of these compounds in a separate BioMAP panel of systems that specifically interrogates fibrosis-related disease biology, and then rank the drugs based on their overall IPF potential. So this is a profile of pirfenidone in our BioMAP Diversity Plus panel, which covers a broad range of primary cell biology. Let me take a moment to walk you through the profile, as there's quite a lot of data here. Pirfenidone was profiled at 4 different concentrations, denoted by the different colored lines, in a total of 12 BioMAP systems, which are denoted by these icons on top and separated by the vertical lines. These systems cover broad disease biology, including vascular, epithelial, and smooth muscle inflammation, as well as immune cell biology and matrix-related biology such as tissue remodeling. Drug effects on 148 readouts are measured across the 12 systems and displayed as a ratioed value relative to a relevant vehicle control that was run in the same assay. We annotate activities on the plot if they meet a defined set of criteria, and this gray shaded area here in the middle is a significance envelope generated from historical vehicle control data and is one of the components used to determine active readouts. So as I previously mentioned, pirfenidone's exact target or targets are unknown. What this profile tells us is that pirfenidone has anti-inflammatory effects, particularly on immune cells. Pirfenidone also suppresses the proliferation of B and T cells, and we also see inhibitory effects on matrix remodeling biology, including decreased production of extracellular matrix components such as fibrillar collagens, matrix metalloproteases, and PI1. Many of these activities are consistent with what has been previously published for pirfenidone. So this is the profile of nintedanib run in the same panel. Nintedanib is known to target multiple tyrosine kinases, and its pleiotropic effects are reflected in this profile. So similar to pirfenidone, nintedanib shares anti-inflammatory and immune suppressive effects in systems containing immune cells, as well as decreased expression of extracellular matrix components. However, in addition to these activities, nintedanib also inhibits the proliferation of a broader range of non-immune cells. Nintedanib has also been approved for the treatment of non-small cell lung cancer, and these broad anti-proliferative activities are consistent with the oncology indication. Nintedanib also modulates tissue factor and thrombomodulin, factors involved in the coagulation cascade, which plays a role in angiogenesis, also consistent with oncology indications. When we overlay the profile of the two molecules together with nintedanib in pink and pirfenidone in black, we identified a number of shared activities which are annotated on the plots. Many of these shared activities have previously been associated with clinical outcome in IPF in published studies, while others have not. We took these shared activities and turned them into a disease signature scorecard, which we use to score the profiles in our database in order to identify compounds with similar effects. Our scoring algorithm was very simplistic. Most readouts were assigned a score of 1, with certain readouts that are well known to be associated with IPF clinical activity being more heavily weighted with a score of 2. Now you can imagine that we could have expanded our initial scoring algorithm to penalize profiles that have a phenotypic signature associated with clinical adverse effects, but for this first pass, we wanted to keep everything very straightforward. Then scored all of the profiles in our database and ranked the corresponding compounds by score. This table shows some of the top scores from the database. The first thing that I would like to point out is that this list includes inhibitors and agonists covering multiple target classes, indicating that there are many potential routes to clinical efficacy. Interestingly, gildanomycin and radicocol, both HSP90 inhibitors, were the top scoring compounds in the database. However, these compounds are known to have toxicity and or stability-related issues and would therefore not be suitable for repurposing. We also noticed that multiple SRC-ABL inhibitors were among the top scoring compounds, which indicated to us that there was something special about this particular target class. Many of these compounds have been clinically approved. The SRC and ABL are structurally similar non-receptor tyrosine kinases that play broad roles in cellular signaling. The SRC family of kinases is composed of 9 members, and most SRC-ABL inhibitors inhibit more than one member of the SRC family to varying degrees. SRC activity has also been associated with the pathogenesis of solid tumors, and recent literature has begun to suggest that SRC may play a role in regulating the differentiation and activation of myofibroblasts, the key pathogenic driver in fibrotic diseases such as IPF. This is a kinome scan tree spot profiler for dasatinib. Each of these spots indicates an individual kinase that is being hit by dasatinib. The size of the spot corresponds to the percent inhibition at a particular concentration, and SRC-ABL-related kinases are colored in blue, while all other kinase hits are colored in red. And what this is showing you is that dasatinib hits a number of other tyrosine kinases as well. The SRC and ABL-related inhibitors have varying degrees of polypharmacology. Shown here are the tree spot visualizations for 4 different inhibitors, from most selective to most polypharmacologic. Corresponding analysis of their IPF potential score suggests that while some polypharmacology may be beneficial, too much may be detrimental. By combining this type of target-based analysis together with phenotypic profiling, we can begin to identify the optimal target selectivity for balancing safety and efficacy in IPF. We then confirm the anti-fibrotic potential of a number of SRC-ABL inhibitors in a more focused BioMAP panel covering fibrosis-relevant biology. This panel consists of 3 systems. These systems cover general myofibroblast activation biology and also include systems containing co-cultures of epithelial cells and fibroblasts to cover physiologically relevant organ-specific biology. The system on the left is a co-culture of small airway epithelial cells and fibroblasts to model pulmonary fibrosis, and the system on the right is a co-culture of renal tubule epithelial cells and fibroblasts to model renal fibrosis. This allowed us to evaluate the anti-fibrotic potential of these agents in different tissue contexts. What this data shows us is that this inhibitor, which is theracatinib, has pleiotropic effects. So we see a reduction in IL-6 and IL-8 expression, indicating anti-inflammatory activity. And we also see some very interesting context-specific effects on matrix-related readouts. So for example, theracatinib inhibits the expression of alpha-smooth muscle actin, which is a key biomarker of myofibroblast activation, in the fibroblast monoculture, as well as the renal epithelial cell fibroblast co-culture. However, there was no effect on alpha-smooth muscle actin expression in the SAE-MyOF system, modeling pulmonary fibrosis. Likewise, the expression of collagens is decreased in the myofibroblast monoculture and the renal tubule epithelial cell fibroblast co-culture, but not in the system modeling pulmonary fibrosis, so we see the same thing. When we compared the profile of theracatinib in this panel to one of the 2 approved IPF drugs, pirfenidone, we see some interesting things here as well. So in the monoculture and the co-culture modeling renal fibrosis, there are a number of shared activities which are annotated on the plot. However, in the system modeling pulmonary fibrosis, the profile is much less similar. So this context-dependent information can also be used to further refine indication selection. So here are the profiles of 4 SRC-ABL-related inhibitors in the fibrosis-relevant BioMAP systems. So what we can see here is that even though these inhibitors are in the same target class, each compound has a unique profile, likely based on their relative target selectivity. So for example, we see that bosutinib and imatinib increase expression of IL-6 and IL-8 in the system modeling renal fibrosis, but that theracatinib and dasatinib do not. Theracatinib increases the expression of pro-inflammatory chemokines such as MCP1, as well as the adhesion molecule VCAM1 in the system modeling pulmonary fibrosis, and dasatinib is the only compound that can decrease expression of alpha-smooth muscle actin and collagen, which is similar to the effects seen for the approved drugs pirfenidone and nintedanib. So we then ranked the SRC-ABL-related inhibitors based on the relative IPF potential score in the wider panel, as well as the number of shared biomarkers with pirfenidone and nintedanib in the pulmonary fibrosis specific panel. So based on this analysis, we now rank dasatinib as a SRC-ABL inhibitor with the most potential for IPF. Interestingly, dasatinib has recently been shown to be effective in a mouse model of pulmonary fibrosis, as well as able to directly block myofibroblast differentiation. So this Venn diagram shows the overlap in the kinase target profiles of bosutinib, dasatinib, and one of the approved IPF drugs, nintedanib. Interestingly, all three molecules are able to inhibit SRC and ABL-related kinases. However, nintedanib and dasatinib, which was a top-ranked hit in our analysis, showed additional activities that weren't shared with the lower-ranking bosutinib. This included DDR1 and DDR2. These are collagen receptors that have been shown to control MMP expression. In addition, both molecules have been shown to inhibit c-KIT, and c-KIT-positive cells in the lung have been shown to promote myofibroblast differentiation. So this information may point to additional targets that contribute to efficacy in anti-fibrotic therapy. So to summarize, we've used phenotypic profiling to define the shared characteristics of approved IPF drugs despite having different targets. We use this phenotypic fingerprint to search a database in order to identify drugs with similar activities. And using this method, we identified SRC and ABL-related inhibitors as having anti-fibrotic potential, which was confirmed in assays more specifically modeling fibrosis. We were then able to rank the hits based on their overall potential specifically for pulmonary fibrosis. So one of the biggest challenges for a drug repurposing program is prioritization of potential indications or prioritization of potential drugs if you have a library. Most companies do have a library of drugs that have gone through some clinical testing and have been shelved for any multitude of reasons. But given an emerging disease indication space such as IPF, how can one quickly identify compounds from that library that might be appropriate for that particular indication? So we all realize by now that for many diseases, there's more than one way to clinical efficacy. And there is emerging evidence that polypharmacologic solutions may be more impactful for certain diseases. So by combining data from human primary cell-based phenotypic screens with data from target-based approaches, one can begin to identify beneficial pharmacology. That brings me to the end of the presentation, and I'm happy to take any questions.
Okay, we've actually already received a couple questions. So if you just give us a moment, we will, our audio will reconnect. Just hold on 1 second. Okay, so we did receive one question. And so this is a question that we commonly get with a lot of our BioMAP systems because we use human primary-derived cells. So how do you account for donor variability of these primary cells in our BioMAP systems?
So that's a great question, and of course for any kind of platform, assay-to-assay reproducibility is important. So to address the question of donor variability, one of the ways that we get around this is by actually pooling primary cells from multiple donors. We have found that this helps to minimize donor-specific effects from run to run.
Thank you, Charlene. And I think we are receiving a couple more questions. So one of the things that is very interesting, we've received this from a couple of our attendees today. Because we are talking about repurposing as well as leveraging this database that BioMAP has, can the customer or can the end users access or know about what kind of drugs that are in the BioMAP database?
So our database does cover drugs that cover a wide range of sort of target biology and disease biology. And while we don't have the information readily available, we can certainly work with any client who has those specific questions. This example is great for clients, is a great example for clients who have their own library of compounds that they're interested in for repurposing for other indications. And so the primary example of this would be for that purpose.
And normally with our BioMAP systems, we don't necessarily disclose the contents of our database, but I believe the BioMAP scientist team has compiled a list of recommended benchmarks. And I believe these recommended benchmarks should be able to give us a good idea, correct, Charlene, of what kind of drugs are active in our systems and what kind of, target biologies that are covered by these drugs.
Absolutely, and I think that's available, I believe, on our website. And also part of any BioMAP profiling project does give you access into our database, in that we do do a database search of a comparison of your particular test agent to our database, and that also is very informative for sort of linking phenotypic fingerprint to mechanism of action.
Thank you. And we received another question regarding actually the if there's a template or if there is kind of a sample report that any of our end users can access. And if you can kindly email us, we would be happy to send a sample report, and that would probably give a very good idea of what kind of data that you can expect as a deliverable. And I think one thing that's very unique about DiscoverX BioMAP offerings is not only do you get the study report, but we will have a scientist, a BioMAP scientist, spend some time discussing the data, and it will be more of a consultative discussion about how to interpret the data and any potential next steps for your research and development programs. So we do have a question about the ethnicities of our donors. Generally, we use healthy donors for our cells and our systems. As you can probably guess, if we use patient cells, there's a lot of donor-donor variability that happens. And BioMAP is actually sensitive enough that they will also pick up on prior drugs that may have been treated with for these patients, which obviously would make the data very, very difficult to interpret. As far as the ethnicities of these primary cells, generally we procure the cells from California. So they're procured locally to BioMAP. So California, as you may imagine, is not restricted to just Americans, but generally we have a fairly good mix of ethnicities, I would say. Please correct me if I'm wrong.
That's right.
And so it's a fairly good mixture, but we do not have anything specific for ethnicities. And I think one of the things that you'll realize from the biomarkers that we use to read out to make these profiles, the biomarkers tend to be very generalized. I should say a population, I should say it's more specifically targeted for population profiling than necessarily individuals or individual ethnicities. So I think we have a very, very good mix. And yeah, we've been in business for a good part of 15 years. So.
Yeah, I mean, I think on the note of, the biomarkers that are part of the BioMAP platform, these biomarkers were chosen because they do have a robust signal in human primary cells, so therefore they are clinically relevant. And they were also chosen, we have many soluble markers or markers that would be easy to translate into a clinical trial to look at drug responsiveness. So these have all been selected to be biologically relevant, to be clinically translatable, and to be robust and reproducible. So that's how we select and narrow it down to, in our Diversity Plus panel, at least 148 different readouts.
Okay, I think there are no more questions, so hopefully we're able to answer all of your questions that you had. Thank you very much for spending the time today to attend our webinar. And Charlene, thank you very much for your time.
Of course, you're welcome.
And if you have any questions, please don't hesitate to contact us at DiscoverX. And we look forward to hearing from you, and hopefully we can work together to build better drugs quicker. Okay, thank you very much.